Hello, everyone. It's Mr. Vallejo. How are you today? Fine, I hope. Today we are taking a look at the cell membrane and diffusion and osmosis. So let's get started and share that screen with you. Today, cell membranes and diffusion. So remember, these PowerPoint slides are available for you in your learning management system, whether that is Canvas or Schoology or Edmodo. Okay, cell membranes and diffusion. Okay, in the first part of today's talk, I'd like to take a look at the structure and function of the cell membrane. Remember, structure and function is one of those topics that is an underlying theme in all of biology. Sometimes we call that an umbrella theme because it covers all of biology. You take a look at the structure of something and understand the structure, you'll better understand the function of that object. So the cell membrane structure and function, understand that the membranes organize the cell activities of the cell, not just the cell membrane that's shown here, but there's smaller pieces within the cells that we studied last time called organelle, and those organelle have membranes also, and those membranes organize the cell's chemical activities. This is where we're headed for the first part of the talk. I'm going to take a look at the structure and show you the phospholipid bilayer. We'll also see that within that bilayer are scattered proteins that have different jobs. And when we put that all together, we get what's called the fluid mosaic model. Before we leave part one, we'll take a look at how some things get into the cell membrane, get into the cell through the cell membrane. Alrighty, this might look a little familiar to you. If you said this is a lipid, then good for you. Remember, a lipid is made of the monomers. It has a glycerol and 3 fatty acids. Here's one fatty acid. Here's another fatty acid. The third fatty acid in this case is replaced by this phosphate group right here. And so this results in not just your average lipid, but is now a phospholipid. The phospholipid has 2 parts. It has a hydrophilic head, that's represented by this gray structure right here, and a hydrophobic tail. Sometimes we draw it like this. Sometimes if I'm on a whiteboard, I would just draw one squiggly line here to represent the fatty acid. So the head is a hydrophilic. Now hydro, you may know, is a prefix that means water. Philic means love. So philic, like Philadelphia, is a city, a brotherly love. Okay, so the hydrophilic heads then are attracted to water, and anthropomorphically, they're attracted to and attached to water. And the tails, conversely, are hydrophobic. Hydro again means water. Phobic means fear of, like you may have a phobia, like arachnophobia or something like that, which is a fear of spiders. So hydrophobic tails, these guys are going to move away from the water or arrange themselves in such a way that they minimize their exposure to water. We saw that in our study of water previously, and these phospholipids then are the main structural component of membranes. But because it has a charged head, a hydrophilic head, and a tail that is nonpolar, then what happens is the phospholipids arrange themselves in a bilayer or a 2-layer sheet with those hydrophilic heads outside. And in this picture, in this orientation, the water here is outside the cell, and this represents water that is inside of the cell. So we have hydrophilic heads here and here, and the tails arranging themselves in such a way that they minimize their exposure to water. We call those hydrophobic interactions, and that's what those tails are doing. Now, scattered amongst the phospholipids, we have different types of proteins. It says here, proteins make the membrane a mosaic of function. Transmembrane protein is one type of protein. It's also called an intrinsic or transport protein. Transmembrane proteins, the prefix trans, we also see from history in the Transcontinental Railroad. If you recall your fourth grade history lessons, the settlers were building a railroad from Missouri out to California and Oregon, and it took them a long time. So some other guys went the other way. They started in San Francisco and went through California up the Sierra Nevadas and went to meet them. And history tells us that they met in Utah near a place called Promontory Point, Utah. And the story is that they took a golden spike to unite those two railroads, one going east and one going west. And when that happened, that railroad went all the way across the continent. So that's why it's called the Transcontinental Railroad. So trans means across. So these membranes are going all the way across. Check that. These proteins are going all the way across the membrane. So with the transmembrane proteins, also called intrinsic or transport proteins, they're called transport proteins when they actually transport something. This one right here has a pore inside of it, and these chemicals represented by these blue spheres are going straight through. The transmembrane proteins can also have other jobs. They can work as enzymes. They can work as receptor sites for signals. So there's just some bigger pictures on those. Here's transport. Going to move these objects across this membrane. These objects cannot go through the whole membrane. They might be charged or they might be too large. So they would only be able to go through the membrane through this pore that's made by this protein. So it's a transport protein. Sometimes these proteins are embedded in the membrane and act as enzymes. We'll learn in a few talks that enzymes are chemicals that lower the energy required to start a chemical reaction. So enzymes make chemical reactions occur that way. And this picture is showing us that these chemicals are going to fit exactly in this three-dimensional shape right here. And when that happens, then the chemical reaction can occur. Another type of protein is called the cytoskeleton protein, and the cytoskeleton protein is a place where the cytoskeleton can attach. In this picture right here, what we see is these proteins are on the inside of the cell membrane. This is it's a stylized version of the drawing we saw earlier with the cell membrane. These are the phospholipids, and these are the transmembrane proteins going all the way across. These are the cytoskeleton proteins, and they're only on the inside, and they serve as an attachment point for the microtubules and the microfilaments that make up the cytoskeletons. If you have a protein that's visible from the outside, those are called exterior proteins. And they're also called extrinsic receptor recognition proteins. However, exterior proteins, their main job, their role is cell-to-cell communication. When two cells get close together, they can talk to each other with the exterior proteins. And normal cells will stop growing when they're touching another cell. Now, cancer cells will continue to grow. Something went wrong with their exterior proteins, and so the cancer cells continue to grow, and they keep growing and growing, and there's no place to grow, so they just start piling on top of each other, and there's still no place to go as they continue to grow, and so they start to break off and go to other pieces. When they clump up, that's called a cyst, and when they break off, we say that the cancer cell has metastasized. That is not a good situation. So here are the exterior proteins acting as a chemical receptor for this chemical messenger. And so here's this chemical messenger, might fit into a site on this protein, and that might be the reaction, the signal to continue some more chemical reactions. And so this is another way for cells to communicate with each other. And still another way is through what we call glycolipids and glycoproteins. Glyco, remember from our inorganic chemistry and chemistry studies, glyco means a carbohydrate. And so what we see here, there are chains of carbohydrates that are either attached to lipids, like this guy right here, or they're short chains that are attached to proteins, like these guys right here. So their role is also cell-to-cell communication. And actually some cancer research, going back to cancer, from a colleague of mine at a local university, what they would do is they would take these oligosaccharides, these short carbohydrates, and take some of the saccharide units off to try to figure out what cancer is doing. And it turned out that you can take some cells and break off a few carbohydrates, and then they lose the recognition, they lose the ability to recognize other cells. So perhaps cancer cells have an issue in their glycolipids and glycoproteins because they can no longer communicate with each other. Now, when we put all this together, this is called the fluid mosaic model. It's fluid and it's mosaic, obviously. Fluid, like milk, it flows and it goes back and forth and it's dynamic and it keeps changing and it's not static. And it's a mosaic in that if you put it all together, it actually does something. This is a mosaic with that's made up of, I don't know, looks like a little colored stipples. And my mom's church, they did something like this on the back wall. They imported different colored bricks, not bricks, but stones from Italy, they told us, and they glued them to the back wall. And when you look at it, it looks like a bunch of bricks. But when you go all the way to the back of the church and look at that wall, you see an image, and that image you see an artful way to see the Virgin Mary and the Jesus child. All right, so here's another picture of the fluid mosaic model. This is all those lipids, those phospholipids, and then these proteins scattered about. So that's why we say it's a fluid mosaic model, which was proposed by Sanger and Nicholson in the 50s, and I think they won the Nobel Prize for that. It's quite a discovery. Before we leave this first part of the talk, there's different types of permeability. Permeability is the ability to have things move through there. So it says here selective permeability. Selective permeability is when you control the flow of substances into or out of the cell. That's what the cell membrane does. This is outside the cell. This is the cytoplasm. Here you can see the cell membrane. These are actually a little bit darker is the phospholipid heads, the hydrophilic heads, and then the space in between, you can't see the tails, but that's what's going on right there. Those are the hydrophobic tails that are together. So at 200,000 power, you can not even see the tails, but you can see the space that those tails take up. Now, there's another term called semi-permeability. Selective permeability happens when the cell membrane selects what goes through. And so there are chemicals that, even though they're the right size, even though they can fit through the hole, they're not selected to go through. So it's like a bouncer at a bar, if you will. Maybe it's a 21 and over bar, and people who are not of age get carded and they can't go in. However, there's another process that goes on called semi-permeability. And in this case here, these blue guys are having no trouble going right through, but these squares here, these orange, beige squares can't go through. Why? I like to say, if it's too wide, it doesn't get inside. So these guys are too big, and so this is semi-permeability. It's the cell membrane has the ability to take in some things, but not things that are too large. They can't fit through that hole. So if it's too wide, it doesn't get. Now, how substances cross the cell membrane is the topic of the second part of this presentation. So I'm going to define diffusion and a special type of diffusion called osmosis. Then we'll take a look at some sets of terms that would be useful to know. We'll see bulk passage, and then I'll go over with you the differences between simple diffusion, facilitated transport, and active transport. So let's take a look at these different topics. First of all, diffusion. Diffusion is the net movement from high concentration to low concentration. Now, in my teacher's manual for this part, it will often say, take a can of body spray and spray it in your classroom. And I don't like to do that anymore because the person in the first two rows, they get sprayed and knocked out, whereas the person at the very back of my room won't smell it for quite some time. That's because it goes from a high concentration of scent molecules in the can, and then once you release that button, then it spreads out. So that's a movement from high concentration to low concentration. Now we say it's a net movement because one of those scent molecules doesn't have to go completely away from the can. It can go back and forth and side to side, but the net movement, the overall movement, is that those scent molecules are moving out away from the can. Now the rate of this movement is greater in different situations, and so we see that there's something called the concentration gradient. The concentration gradient, it says here, it's a difference in concentration between two different areas. It says steeper is faster. On my way to one of my online colleges, I finished teaching in one place at 2:30. And I've got to get to my online college, my other college at 4 o'clock, and so it's a good 30-minute drive, and so sometimes I really got to hustle to get from one place to another. But when I take the freeway, I've got to go up a hill, and coming down the hill on the other side, it's a little spooky because you take your foot off the accelerator, and you still continue to move faster and faster. So that's a pretty steep hill. So it has a pretty steep grade, we call it. So if you look at this term here, concentration gradient, it has to do with the steepness of the difference between the top and the bottom. So when you have a concentration gradient, if you have a difference between inside and outside, that's very different, then you will have a greater flow, a steeper hill, if you will. This is a really steep hill in San Francisco, and maybe some of you have been on. And it's a touristy thing. So steeper is faster. Now, specifically, we take a look at osmosis. Osmosis is a special kind of diffusion that has to do with water. And not only just water, but water through a membrane, a semi-permeable membrane. In this picture right here, these water molecules are blue spheres right here. And these green things right here, these are sugar molecules. These are sucrose molecules. Now, the membrane is too large. Now, the sucrose molecules are too large to go through the membrane. So that's why they're stuck on the right side. But the water molecules can go back and forth. And you can see that from these black arrows, they're able to go back and forth. But the net movement is from the left to the right. That's because the gradient is such that there's 100% water molecules on this side. And it might be, 90% water on this side. So the gradient is still 100 to 90. And so the flow goes from highest to lowest. And so that is osmosis. Osmosis is diffusion of water through a semi-permeable membrane. And we can see here's a classic experiment with a U-tube, and then you can see over here a similar picture. Just this time the water molecules are red with the oxygen and two hydrogens that are white attached. And you can see that they're going back and forth through this membrane. And this molecule here is says a solute molecule with a cluster of water molecules. And when that happens, and these water molecules are going over here, then you get a movement in this tube. So that's an experiment that shows us the processes of osmosis. But really what I'd like you to know is I'd like you to know these terms: hypertonic, hypotonic, and isotonic. Hyper means a lot of. I have a lot of energy. There's some people say little kids get a sugar high. And so back in the day, we'd say that, oh, your kid is hyperactive. And we don't use that term anymore because some people actually do have a condition called hyperactivity. But a hyper means a lot of. Hypo, on the other hand, means you have a little bit, like hypoglycemia when your blood sugar is low. Isotonic, we saw this in the chemistry lecture. Iso means the same. So iso, same, hypo, a little, hyper, a lot. Tonic, now tonic, if you think of mineral water, think of tonic water. I remember when I was 12, I won a case of tonic water in a radio contest. My friend Mike and I rode our bikes 4 miles to get to the radio station. They gave us our 6-pack of Canada Dry tonic water. And we went back to our bikes and popped the lid on those free drinks and started to guzzle. Oh, it was horrible. Because if you've never had tonic water by itself, it tastes like dirty water. And tonic means dissolved stuff. So hypertonic means you have a lot of dissolved objects in there, very salty water. Hypotonic means you don't have enough. So hypotonic solution might be very close to distilled water. And isotonic solution is one that's the same. And so in these pictures right here, here's a hypertonic solution. A red blood cell would shrivel up in a hypertonic solution. Here's a hypotonic solution in distilled water. A plant cell would get harder and harder as the water goes in here and pushes it up against the cell wall of the plant cell. And then in this isotonic solution, you have water going in and water going out. And it's the same outside as it is inside. So that's what we call an isotonic solution. Now here's some terms that would be useful for you to know as we study diffusion and osmosis and the structure and function of the cell membrane. Here is an earthworm. An earthworm is an annelid that does not have any bones, but it does have a skeleton. We say that it has a hydrostatic skeleton. What that means is a liquid, the body fluids inside that worm are pushing up against the skin, and that's what holds it together. So that pressure of the liquid, the body liquids going against that skin, is called osmotic pressure. So we have a hydrostatic skeleton, which is being maintained by osmotic pressure. This is a cell, a picture of a cell that's going through, that is turgid, we say. And it is turgid because it has a lot of turgor pressure or osmotic pressure. And if there's too much pressure, you're going to have a situation called extrusion. I once was sitting underneath the tree. I was lucky to get a seat underneath the tree because it was a hot day and I would have liked the shade. I saw some people get up, so I grabbed the bench, I sat on the bench. A few minutes later, I got up as well. Why? Because I looked up and there was liquid dripping on me on a hot day. It wasn't raining, and what was that? What it was a tree was going through extrusion, and it was getting so much water up its stem and up to the leaves that the leaves didn't know what to do with that water, and the pressure of the water, it was such that it squirted off the very tips of the leaves. It felt like a little like it was raining. So that's kind of an odd situation. That's called extrusion. Here's some LOD cells that are exhibiting plasmolysis. They're in a hypertonic solution, like a 30% salt water solution. And these guys are shrinking away. This is the cell membrane, which you don't usually see separate from the cell wall. The cell wall is right here. The cell membrane is right here. And in the opposite situation, in a hypotonic solution, this green is going to be right up against the cell wall. So that's called plasmolysis when that happens. All right. So let's take a look at bulk passage. Bulk passage occurs when you have a lot of objects going in at once. I shop at Costco. We buy bulk. You get a lot of it at once. Some people don't like a lot of toilet paper. I have 7 kids, so we like getting a lot of toilet paper at once. But if you get it all at once, that's called buying in bulk. So the cell sometimes gets rid of things in bulk. And so what's happening here is these purple guys right here are in a vesicle on their way out of the cell. It might be that this is a protein that's meant to be secreted. So the secretory protein is in a vesicle, and the vesicle fuses with the cell membrane, which releases the protein to be secreted, and also increases the size of the cell wall right here. So that's how the cell gets bigger. The opposite process is called endocytosis, and endocytosis is when you gobble up some of these guys all at once, and sometimes I call it cellular eating. These membranes fold inward and enclose materials from the outside, and you get this vesicle. What may happen is another vesicle may come over and join with that, and like a lysosome that has lysozymes in it, and it'll break down these things right there. Specific types of endocytoses are phagocytoses and pinocytoses. Phagocytoses we call cellular eating. Pinocytosis is cellular drinking. You see here are examples. There's an amoeba that is eating something, probably an algae or something, and it's engulfing it, and so it's cell eating. And these little arrows were added to this picture right here to help us see the pinocytosis that is occurring in that picture. Now, probably one of the most important topics in this talk is the difference between simple diffusion, facilitated transport, and active transport. So for each of these processes, we're going to need you to know whether it requires energy, whether it requires a protein, and which way the gradient goes. Is it from high to low, or does it go from low to high? So for simple diffusion, we see those 3 points right there. No extra energy is required to do simple diffusion, because over here you have these green guys, and then you have this membrane. And what's happening is that these green guys will settle themselves apart so they're equal on both sides. Here you start out with 12 greens on the left. At the end, at equilibrium, that's 6 on each side. Even if you got these 6 red ones on the right over here, throw those in the mix, you're going to have 3 and 3 by the time it's all done. So no extra energy is required, and these guys move from high concentration to low concentration, and it does not need the help of a protein, so no proteins required. Simple diffusion we see with small nonpolar molecules such as oxygen or carbon dioxide, and these guys can easily pass through the phospholipids that make up the phospholipid bilayer. All right, here's a picture of a guy with a tie. Some people say never trust a guy with a tie. Here's another guy with a tie. Well, these guys are facilitators. I remember going to a meeting once, and the guy said, you know what? whatever happens in this meeting is going to happen. I'm only the facilitator. I'm going to help what's going to happen. I'm going to help it happen quicker. So that's what a facilitator does. The rate is faster than expected, so it must be helped. In this case, by the facilitator. In the cell, it's by protein. So a protein is required. It still goes from high concentration to low concentration. You see these yellow guys, a lot of them, moving towards where there's just a few of these yellow guys down here inside the cell. So it's still going from high concentration to low concentration through this protein, but no extra energy is required. Now that's different from what we call active transport. The classic example of active transport is the sodium potassium pump. And what happens in active transport is that we have these guys, we have 4 of these on the outside, 2 on the inside, but that doesn't matter. You're gonna pump these guys out. You're gonna go against the gradient. So you're going opposite of what it's like going uphill. So because you're going uphill, you're gonna require extra energy and you're gonna need some help. And so it's these transport proteins right here, which provide the help for the active transport. So this is another way to get things in and out. Classically, we see this happening in your muscles and in your nervous system. They take advantage of active transport of electrolytes going back and forth. Here's a term right here I need you to know. This is phosphorylation. And what happens in phosphorylation is you see the movement of that phosphate group. There's 3 phosphates attached to this adenosine. That's what we call these adenosine triphosphate, or ATP. And so when we move this phosphate off, what you have is the resulting, everything else is adenosine diphosphate for 2, but this one phosphate comes and it does something else. In this case, it attaches to the protein and causes a change in shape of the protein. And so in this particular example, then when this protein changes shape, it releases those guys, the solute to the environment. So phosphorylation is the movement of a phosphate group. All righty. Here's some practical examples, but we're running out of time here, so I will leave you with this. If you really understand what's going on today with this last section, you'll be able to correctly fill out what we call the semantics features analysis grid. For those of you who are education majors, but if you're not in education, here's a table for you to fill out. And so what we want you to know is which way does this concentration gradient go? Does it need a protein? Does it need extra energy? Well, to give you a hint, for the concentration gradient, for facilitated diffusion, it goes from high to low. You know what these guys are? Hopefully you can fill those in. Does it need a protein? Well, simple diffusion doesn't need a protein. Does active transport need extra energy? Sure does. Now, if you can fill out the other ones, you're doing really well, and you're on your way to understanding diffusion, osmosis, and the structure and function of the cell membrane. So we'll see you next time. I'm Mr. Vallejo. Remember, biology is fun.